In this session, let us discuss about gene therapy. Dear students, what is gene therapy? Gene therapy, which is a type of treatment in which the treatment gives to genes or a target genes, means it is a technique that modifies a person's genes to treat or cure diseases. Or gene therapy is a technique which involves the replacement of defective genes with healthy ones in order to treat genetic disorders. And this gene therapy is an artificial method that introduces DNA into cells of the human body. The first gene therapy was successfully accomplished in the year 1989. So, in general, gene therapy means a type of treatment where the person's genes are modified to treat the various diseases by using the technique. See, this diagram shows a very simple process of gene therapy. The first diagram, this is a cell with a non-functioning gene. Into this non-functioning cell with non-functioning gene, into this, in the second step, they are going to add DNA containing functional version of the lost gene. So here, this non-functioning cell with non-functioning becomes active, and now it is the functioning normally. This is very simple technique and process which represents the diagram. Diagram represents very simple process of gene therapy. So there are two types of gene therapy. One is somatic gene therapy and germline or germ cells gene therapy. So the name itself says somatic gene therapy. In somatic gene therapy technique, here the body cells of the patients are targeted for the foreign gene transfer. In this case, the effects caused by the foreign gene is restricted to the individual patients only. It is not inherited by the patients, offspring or later generations. It is only individual treatment or patient treatment. That is called somatic gene therapy. Whereas second type is germ cells gene therapy or germline gene therapy. Here the therapy is going to give to the germ cells, and the genes are inserted into the genomes or the genomes are inserted into the germ cells, means reproductive cells, that is sperm or eggs or ovum. So targeting of germ cells makes the therapy inheritable, means so by giving the therapy to the germ cells or reproductive cells, the therapy is inheritable, making the therapy is heritable. Okay. So this is about somatic gene therapy and germline gene therapy. These are the two major types which are used to treat the genes. And there are some vectors which are used in the gene therapy technique. They are viral vectors and non-viral vectors. See, viral vectors means here the viruses are becoming the vectors to transfer the genome into the targeted genes. Here some of the retroviruses, adenoviruses and adeno-associated viruses, AAV, adeno-associated viruses, are commonly used viral vectors, whereas some of the less commonly used viral vectors are derived from herpes simplex virus, means HSV1. These are the viral vectors which are used in the gene therapy technique. And there are some non-viral vectors which are also used in the gene therapy technique. They are non-viral vectors includes chemicals and physical methods such as direct injection of plasmid DNA into the body or cells, and receptor mediated endocytosis, and the gene transfer through liposomes, polymers, nanoparticles, etc. These are all some of the vectors which are used in gene therapy technique. They are non-viral vectors. So vectors are both viral vectors and non-viral vectors. Okay. Now this is one of the diagram which clearly represents, that is, adenovirus as a vector in gene therapy. So here this adenovirus used as a vector to transfer the genes into the target organism or organism's cells DNA. Okay, so the picture clearly represents here the vector insert into the cell, and then the virus vector split or discharges the DNA into the target cell. So that is adenovirus as a vector in gene therapy. This is one best example. Like this, all vectors are used in the technique of gene therapy. So there are two types of methods in gene therapy, that is ex vivo gene therapy and in vivo gene therapy. The name itself says ex vivo means the outside the body. The genes are transferred into the patient cell. Then in vivo gene therapy means it transfers the genes directly into the cells inside the body. That is known as ex vivo gene therapy and in vivo gene therapy. See here, this picture clearly represents in vivo gene therapy and ex vivo gene therapy. So here in vivo gene therapy, vectors with a healthy gene, vectors are directly inserted into the cells inside the body, whereas here the cells are taken out from the host which are defective genes, and the vectors are through the vectors into the defective cells, the genes are transferred, and later host cells with the healthy gene later inserted or introduced into the human body. This is about ex vivo gene therapy, and this is about in vivo gene therapy. This totally picture represents clearly both the types or both the methods of gene therapy, in vivo and ex vivo. And there are some applications of gene therapy. As I told earlier, gene therapy is used to treat the various diseases or defective genes. Here some of the important applications. So the cancer in gene therapy is used in to treat the cancer, which includes glioblastoma, breast cancer, head and neck cancer, and other cancers like prostate cancer. And gene therapy is also used to treat neurological disorders. Neurological disorders includes Parkinson's disease, Alzheimer's disease. These are the disorders which are known as neurological disorders. To treat these neurological disorders, the gene therapy is very much useful. And it is also useful in treating inherited diseases like hemophilia, cystic fibrosis, severe combined immune deficiencies, SCID. And it also useful in infectious diseases to treat the infectious diseases like tuberculosis, malaria, HIV, and influenza. And gene therapy is also used, applied in case of cardiac diseases. These are some of the few important applications like this to treat the disorders or diseases. Gene therapy is very much useful. Dear students, I hope you all understood in this session about gene therapy and the types of gene therapy and the methods which are used in the gene therapy, and finally applications of gene therapy. I hope you all understood well. Thank you. Thank you one and all.